Okay, I'm gonna quickly try to summarize DNA for you. Sequencing technique is very important that we learn this. DNA sequencing was originally is now really changed a lot, and basically bottom line is there's now machines that do the DNA sequencing. Just throw what you do is you take an unknown piece of DNA, and single strand, and you just throw it in through a very small pore, and then identify the bases one by one by the way there's a distinct change in the electrical current, because obviously each base interacts with a current differently, and so as that DNA moves through the current, it changes the current in this particular way, and then you just read the current readings. Then you can, that's why it's almost how you read the DNA. So you can read the DNA sequence because you have no idea what letters are there, and then you can read the sequence as it travels through this particular nanopore membrane as it interrupts electrical current. And all of this is in figure 20.1. Figure 20.1 here. A technique which a DNA strand is passed through a small pore in the membrane. The resulting changes in electrical current are used to determine the sequence. There's even better methods than this now. They just keep getting better and better, the sequencing. So sequencing has really become an incredible thing. Okay. To improve sequencing techniques to transform the way we do biology, complete genome sequencing. I don't think we really need to worry about too much of the new ways. Softwares can figure it out now. Now AI is being used. So we can come back to this. What you're gonna need to know is what do you do with the gene once it's sequenced, okay? Your job as a scientist is not gonna be to sequence it. That's gonna be the job of the machines. Your job is now to understand the science behind what are you doing with these genes, what do you do with these genes and all this kind of stuff. All right, so let's talk about that. So first of all, it's important to understand that when you have a piece of DNA, only a small portion of the DNA codes for a protein, right? So in many eukaryotic genomes, the protein coding gene only occupies a small portion of the DNA. The rest of it's garbage, non-coding sequences. Literally, a single human gene, for example, might only make up one gene is a 1/100,000th of the actual DNA molecule. So it's like a little thing in this vast thing. And further, what makes this really tricky, it's not easy to distinguish the gene from the surrounding sequences because they all look the same, right? So you got to isolate the specific gene. So isolating a specific little gene and then make copies of it. Okay, so that's the whole idea. So many methods for cloning the DNA have been now produced. So now the way they use it is mostly taking advantage of bacteria, which really replicate fast. And so you can replicate the DNA very fast too, right? Because bacteria grow quickly. And plasmids, you can easily cut the plasmid, which is a circular version of DNA. And then you can cut it and then basically put something inside of here. So we can talk about how we do that. So we take a DNA, a gene, and put it inside this plasmid. We'll show you how we do that. And then we put the plasmid inside the bacteria, and the bacteria is grown and cultured to form tons of these. Okay, now you got tons of genes. And then a protein's even expressed from the particular gene in the bacteria. So your human gene can actually be expressing human protein. Now they have to sometimes it doesn't work so well, but it seems to. Or you take a gene interest and you make tons of copies of the gene. So there's a couple of options. You could either take the gene, make tons of copies of the gene, and then you could put those genes into, say, a particular indication. This, for example, they put it into plants to make them resistant to certain bacteria, or you change the bacteria to do something else. Here, they took it to make tons of protein, and then when they took all the protein made in the bacteria, it's like a hormone, and then they gave the hormone to a kid who is not growing, and he grows, okay? Or they make it a special protein to help dissolve blood clots, like TPA, and that's made inside the TPA is a blood clot dissolving thing. Okay, anyway, these are called recombinant bacteria. Recombinant bacteria is an important time. This whole thing is called recombinant DNA. So whenever you have a DNA that's mixed, it's called recombinant. Either the bacteria is recombinant or DNA is recombinant. That word is really important. What is that? The recombinant it means it's a mix. Molecules from two different sources. Okay, just know that term. You'll see that all the time, and they can produce recombinant DNA, right? So the plasma acts as a cloning vector, okay? And the DNA can carry foreign DNA, widely used because you manipulate these, okay? We'll talk about how we do that. That's what they said here. We talked about that already. Okay, now how do we do it? Really, the whole idea comes from this concept of restriction fragments and restriction sites. It's important to know this. What's a restriction enzyme? And you're going to need, this may be on your test. This may be on your test. This is very important. So first of all, there's a restriction endonuclease or restriction enzyme. This is discovered in the 1960s, even before I was born, by biologists who were doing basic research on bacteria, and they found that these enzymes actually cut up DNA. Now, for example, why is that important? So let's say a virus DNA comes into a bacteria, and the bacteria says, well, shit, I don't want that virus DNA in here. Basically, it's a scissor. It's gonna basically cut it up into pieces so that that viral DNA doesn't work, right? So the restriction enzyme protect the bacteria by cutting up the foreign DNA. Literally, it seizes the foreign DNA, starts cutting up. So now, how does the restriction enzyme know it's a foreign DNA or not its own DNA? How is the restriction enzyme so smart? It's not that smart. It just cuts up anything. What is smart is that bacteria protect itself from being cut up. That they add methyl groups to these adenine and cytosine in their own sequence. So basically, the bacteria protect themselves from their own enzyme. So the enzyme won't destroy their own bacteria. It's just and how does it protect it? Because it's the CH3 methyl group is attached in the bases, and basically now it can't be. So it was an evolutionary development. It's genius, isn't it? Now, next thing. It's important to know what's a restriction. So the restriction enzyme cuts a really specific segment. I mean, to the letter. Like I'm gonna cut only this particular thing. Whenever I see this, that's when I'm gonna cut it. So it can only cut something. So for example, this restriction site is for this enzyme, E-CoRI. Okay, now E-CoRI only cuts at that site, GA. Okay, and then when it cuts at the GA and cuts at the GA, it turns out that now you have the sticky ends. So when it cuts at the GA, you have the sticky ends. And what are sticky ends? Sticky ends are this part, kind of the part that's hanging out here. That's a really important sticky ends. Sticky ends mean that you can use this SNES segment and attach it anywhere else. So now you know what you do now. You go find. Now it's not just a GA, right? It's got to have the same matching sticky end. So now you go throw in your restriction enzyme, the same restriction enzyme, into a human DNA molecule, and it cuts that up the same way. And then hopefully within those two sites, you've captured the DNA you're interested in. And then you bring that in there and then it just kind of sticks in, okay? So that's basically the combo. And it's called recombinant DNA. So the idea is that the DNA is, that's it. So because any sequence that is short by chance many times in long DNA, a restriction enzyme, because any sequence that is this short occurs by chance many times in a DNA molecule. So you'll find this sequence so short, it's probably commonly found as a repeating thing, and then it cuts it up in different places. So then you look for your best fragment, and then you look for your best fragment, different set of restriction fragments, and since restriction enzymes always cut at the same DNA site, copies of DNA molecules exposed to the same restriction enzyme will always see the same fragments. So you can always get the same fragments every time when you cut it up. Okay, so and then you can use those fragments and shove it into the bacteria. Now, most useful restriction to cleave the backbones in a staggered manner. Well, that's the sticky ends. We understand that. Okay, now to check after they've been copied many times to make sure the fragment has been inserted, a researcher might cut the products again using the same restriction enzyme to see to check the recombinant parent after they've been copied many times to make sure the fragment actually has been inserted, right? Meaning that, just because it's sticky end, how do you know it actually stuck? And then actually, did it actually get cloned on the replication, right? And if the insert is there will be two fragments if they throw in a restriction enzyme, meaning that one site from the plasma site and one site from the inserted DNA. So basically, that's so to separate and individualize the fragments, carry out gel electrophoresis. All right, so that's what you use gel electrophoresis for. So basically, what happens is you go backwards this way. So you're going to go in, you're going to cut. Remember that this is your plasmid. You wanted to know about this if it really is there, if this thing is actually there, rather than this, rejoining back together. We want to make sure this managed to sneak in. And if this managed to sneak in, what you're going to do is go re-expose this thing to the enzyme, and he's going to cut this up into this fragment. So you're going to have this thing. Now, what's going to happen is you're going to have two fragments, one with the size of the plasmid, one the size of the inserted DNA, right? So that's going to be the two different fragments, right? There's going to be two different sizes. That's the important thing. There's going to be two fragments. One the size of the inserted DNA, right? And then he's going to cut on this side and he's going to cut on that side. So that's going to be the one fragment. And the other fragment will be this whole thing. Will be this fragment right here, right? This fragment. So that will be fragment 1. This will be fragment 2, right? That's it. Bottom line is you're going to cut here, and you're going to cut here. So you have two fragments, 1 and 2, right? Obviously. So that's the bottom line. And so then so if the insert is there, you're going to have two. So the insert is there, you're going to have two fragments. And then, so that means that how do you know you have two fragments? You use gel electrophoresis, which the shorter fragment, which is usually going to be the insert, will travel faster and help separate the fragment by length. That's typically how it's done then, okay? So how does, and gel electrophoresis kind of works basically, you dump your bacteria in here after you cut it up, and then you turn on this electrical thing, and basically they start swimming. So these guys swim through the gel, and as they swim through the gel, the winner is always the shorter guy. So he always comes to the top, to the closer to the other side, to the positive side, because remember DNA is negative, right? So they travel to the positive anode. So the winner is always going to be the short fragment. And this gives you an idea that what they do is they have a standard size fragment, so depending how far you are in the swimming pool, right? So the shorter one is measure a certain length, that's a certain length, they know this from beforehand. So this is kind of their ruler. So this is their ruler of standard lengths. So if something reaches this point, they say, oh yeah, it matches up, that's exactly the size that we were looking for, or that we know, hey, there's two fragments that this segment lit up and this one, there must be two fragments. So if it was just one fragment, after you cut the DNA, let's say it was a thing, and you just cut the DNA, and then all you see is one fragment, then you can guess that there is no insert. Does that make sense? That you say, hey, there's only one fragment. How come there should be two fragments? So you'd expect two fragments on the gel electrophoresis after you cut it up. Okay, that's the whole point. Okay, now, what is PCR used for? PCR is pretty neat. PCR is neat. It basically allows you to grow a super small amount of DNA. Let's say you had like a you found a little bone, and you think, oh man, that bone belongs to a dinosaur, and all you can get is a little touch of it, but you want to sequence the DNA in that something there, whatever it is. Okay, now all you need is just even a drop of blood, not even a drop of blood, a little blood, even from like to investigation site, and you want to confirm somebody it was there or something. Then you can just take that little piece, and then you can really replicate it. And what's the equation for replication? 2 to the n for each cycle. So if you run the cycle once, you're going to get two nucleotides, right? So why? Because one, you split it, and then you replicate it, okay? And so the replication is basically using a primer and then the DNA polymerase, right? Now it's important, I'll tell you about that, that's really it's cool. And then but if you run it a couple times, then you can really expand it. You should recycle it, right? So first you got to heat it up. So you heat it, and that separates the hydrogen bonds. And then you cool it with primers on each end. And that allows, because you don't know what the sequence is, right? So you have no idea what the sequence is. Now, the tricky part is, how do you get the primer in there? I don't know that part. That's I'm not really sure. But anyway, bottom line is this, is you can grow the bacteria using DNA polymerase. Now, they only use a very specific polymerase called Taq polymerase or PFU polymerase, because it can handle the heat. So as this heating is going on, and this kind of stuff, that polymerase won't die out. So the key to automatic PCR was a discovery of a heat-stable DNA polymerase called Taq polymerase. Otherwise, this wouldn't work. Okay. Okay, I got to get going to school or to hospital. But basically, very, very fast. You need a little bit, grow a lot. But you can't actually use this for cloning, because the Taq polymerase that they use is not really a proofreader. So there could be little mistakes and stuff like that. So basically, you basically get some good copy. Our PCR primers are synthesized to include a restriction site of the fragment. So basically, it's important is if you want to clone it, right? And you want to say, okay, I want to do the proper cloning, and I want to do it cloning the right way, grow it, and not crazy this way, but clone it using bacteria. Then you gotta have restriction sites so you can stick it in the bacteria. So what you do is you actually throw in a restriction site here and here. Okay, so that way once this grows, you can take this that's got the restriction sites on there, and then you can shove that into the DNA molecule. But you got to pick your right bacteria using, I think they use this to kind of help figure out which one you're using. So basically, you basically say, okay, instead of PCR to provide to the specific fragment to clone, the PCR primers are synthesized to include a restriction site at the end of the fragment that matches the site and the cloning vector, and the fragment and the vector are cut and ligated together. The fragment, each end of the DNA fragment matches the site and the cloning vector, all right? So basically the DNA is synthesized to include a restriction site at each end of the fragment. So the primer includes a restriction site. That's this primer includes a restriction site. Okay. Okay. So then, and then that can go into the vector, and then the plasmids can then do the proper cloning without any errors. You know, maybe I think they sequence it. They probably take it, sequence it to make sure that there's no errors, and they double check and make sure there's no errors, and once they figure it out, that kind of thing. Well, anyway, so that's how they figure out that. So you take remember the restriction. So this is the two pieces. Remember, piece 1, piece 2, right? This was the site that you're interested in. So this is cut with restriction and the same enzyme, right? So the primers are designed so that the DNA fragments obtained by the PCR have a restriction site at each end. To the restriction site at each end, right? So restriction site, and then you got your bacterial cells, and you just shove it together and you mix, right? There you go. And only the cells that take up a plasmid will survive. Well, that's just now this is interesting. Recombinant DNA plasmid are added to bacterial host cells that are treated with antibiotic. Only the cells that take up the plasmid will survive due to the antibiotic resistant gene. So there's actually an antibiotic resistant gene that's in here. They kind of trick the cells. They actually throw that in there as a trick. That, okay, did this plasmid actually get into this bacteria? How do you know? There's no way. You can't look at the bacteria and say, oh, is there a plasmid in there? There's no way you know. So what you do is you say, okay, I'm going to throw in my plasmid a little gene that makes the cells resistant to the antibiotic, so the antibiotic can't kill it. So then what happens is that when this gets in here, that goes in here, you have two things. You've got the restriction site. This has nothing to do with anything. This just has the known little gene in there for bacteria resistance. So now when it gets inside of here, antibiotic can't kill this, but it kills these guys. So now the only bacteria that are alive are the guys that have the plasmid in there, and these guys clone. Otherwise, you have so much garbage. The garbage, right? So this cleans it up. Okay. Now, once the gene is in here, you want to express the gene, right? But to express the gene, sometimes you have to kind of overcome some differences between bacteria and human cells when you're mixing this up. And so there's a thing here called an expression vector. So the expression vector actually sits upstream to the restriction site. So you got your restriction site, right? That's a restriction site. So maybe kind of like right there. Oh, sorry. So this is the restriction site. The cloning vector has a promoter. Well, now what's a promoter? So there'll be a promoter here. A promoter is something that always overdrives, or basically stimulates transcription for a protein, right, to make RNA. So you put in a promoter next to the restriction site. So promoter just upstream to a restriction site, and then the bacterial host cell will then recognize the promoter, and then continue to express the foreign gene. So now the promoter's here and it really sees this gene and it starts really kind of turning it on and delivering. And then the other thing that it does is you got to make sure that whenever you put this in here, it's clean, right? Because you don't want to have garbage in here and then put the garbage inside this, and this is like, what is this garbage? So it's got to be clean. It's got to only what they call only exons, because there are non-coding regions. They're called introns, and too many introns can make the gene real long and basically not get the right expression that you're looking for in the bacterial cells. So that's an important thing. So they've been trying to make things a little bit more okay. But now they're getting better. It's okay, well, we don't need to rely on these bacteria. We're going to rely on yeast, because yeast kind of, we're kind of relate to each other. And so now we can there's ways to inject in the yeast and be more specific and that kind of thing. Okay, we'll talk about more of this later, but I want you to, if you have a chance to do over this, I gotta get to the hospital real late. Bye.